All right, so our last presentation in the morning is on the topic of recurrence, which I think all of you at some point have probably at least thought about. And it's a great honor and pleasure for me to introduce today Dr. John Johnson, who, as you know, is a newcomer to this group. I actually first became aware of Dr. Johnson's work because of a very interesting paper that he published a few years ago addressing exactly the issue of a recurrence in a rather through an uncharacteristic mechanism. But I thought that combined with, at one point there was a recent discussion on Facebook among the 11Q families about the issue of recurrence. So I thought it'd be great to have someone who knows genetics far more than I do to discuss the real nuances. I want to just give you a little bit of Dr. Johnson's background. He has been in the field of genetics for over 30 years, with stints at the University of Utah, at Children's Hospital of Oakland, almost 20 years in Montana at Shodair Hospital, and now, most recently for family reasons, took over as Chief of Clinical Genetics and Metabolism at Tufts Medical Center in Boston. However, his proudest achievement has nothing to do with genetics, but it involves his secondary passion, which is music. I tried to get him to bring a keyboard on the plane. It didn't quite work out, but take a listen to this. He played keyboards in the opening band for the group Kansas in Helena, Montana. All right, I'm showing my age. How many of you remember the group Kansas? All right, we're all up there in age. He thinks it would have been more exciting had he told you that a few years ago, but you know, you're in good company here. So it's a great honor and pleasure to have you, and thank you so much for coming. We have a little bottle of wine that has been provided to us from a family who has a private vineyard up north, so make sure you grab one of those. And how about a warm round of applause for Dr. Johnson?
Well, actually, I first came across you about the time I was working on this family. This has been quite a while ago. And sent you a couple emails. They went back and forth, and they're buried somewhere in the late 90s, early 2000s. And the project sort of languished until I finally, we got some new technology in Montana where I was at that point. And I finally finished off this project. So it's been a long road getting here in multiple ways. I guess the other way was getting here yesterday. I spent 5 hours in Dallas, so I pulled in at 3 in the morning my time, so I'm a little spacey, but I'll get there. So I've actually spent, I think if I add it up, I've spent 11 or 12 years in California. I've been to San Diego numerous times. Mostly, spent most of my time in Northern California in the Bay Area, but I was down in LA for a couple of years. And so I've been here a lot, and I really enjoy coming to San Diego. This is not going to be a very long trip. My wife was supposed to have had her hip replaced on Monday, so after I agreed to speak on Monday, I had to move that to today. She then decides not to get her hip done now. I end up in the hospital last weekend. Anyway, you know, that's what life throws at you, I guess, as you well know. You have more experience with some of these life curves than I do, probably. So this is the formal title of this article that I finally put together. And basically, everybody on this page is in Montana when this started, but as you can see, some people have moved around, so I'm now in Boston. Mary Haig was the cytogeneticist, and she's down with the company in New Mexico. And Linda Beischel was the one who actually did a lot of the DNA work, and she now works for the newborn screening program in Montana. Kathy McCann is a genetic counselor at Phoenix, and the rest of the folks are still there, including Dr. Hansen in pediatrics, but Jim Reynolds, James Reynolds, the last one on the list right above Bozeman, just retired. We used to call him General Jim. He did a couple of years in Iraq and in Bosnia, and left me alone both times, so I'm not overly thrilled that he's General Jim, but anyway, he just was able to retire. Okay, so the story is about two different angles on the syndrome. One is chromosomes, and you're pretty familiar with those because that's what you're dealing with. And obviously the chromosome of interest I've got highlighted here. But it's also about DNA. And okay. And basically, DNA is a very interesting molecule. It's like a zipper. So if you have the DNA sequence, which is very simple, it's a four alphabet code. On one strand, you can predict what the other strand will be. You can separate the strands, and they'll find each other in space. It may take a while or whatever, but they will find each other. On the other hand, strands that don't quite match will find a strand that you've separated, and mismatches are tolerated unless they get fairly extensive, and then the two strands will be kind of like a magnet with the two poles being opposite, so they won't come together. So, I don't need to tell you much about Jacobson syndrome, except to focus on those last couple of sentences. So, how many here have two affecteds? Did I hear you said boys?
I have two boys.
Two boys. Anybody else? You have a translocation, okay. And anybody else in the group over the years had two? Okay, yeah, I mean, it's not... Yeah, there are some, yeah, Dr. Grossfeld mentioned a couple. Yeah, mostly they're due to chromosome translocation, and there have been some of those written about, but there are a few that don't have translocation, and that's what I'm going to talk about. So the first thing I need to talk about is translocation. So basically, the syndrome we're talking about is on the end of chromosome 11. I just pulled another chromosome as an example. So translocation is when two chromosomes experience breaks, and our DNA is not perfect. We're exposed to, basically, the worst things we're exposed to are oxygen and sun. And of course, we ought to have those, and food is probably third on the list. But, I mean, you know, there's things in our environment that take a toll on our body. And they take a toll on DNA, and they can break chromosomes. So if we had a translocation, we start with breaks like this. And then basically after the material is exchanged, you have what's called a translocation carrier up at the top. But if that translocation carrier passes on the two chromosomes with the arrows back and forth to each other, and that child's going to have Jacobson syndrome. So the pinker chromosome has a deletion, and that would be the typical Jacobson deletion. But notice that child also has a little extra piece of chromosome 8, the one that was traded. So patients who have Jacobson syndrome from translocation are not pure Jacobson syndrome. They've always got something else going on. So they have a deletion of the Jacobson region, an extra or trisomy of some other region. That can make some differences in what they might look like. So we had I've probably seen no more than 4 of these cases over the year. First one I saw was one of these serendipitous diagnoses when a kid had congenital heart disease and low platelet count in the NICU, and I'm looking at this old French chromosome textbook, and it talks about 11q deletions and thrombocytopenia. People were barely even calling it Jacobson syndrome at that time. So I diagnosed that kid, and then I had another real nice family still in Montana. This family I lost track of, and I don't know if they're part of this group or have been part of this group, but I certainly tried to get hold of them when I wrote the article up, but I lost touch with them. So anyway, the first guy was born, and he's on the left here, and you don't have to read all the details or understand them really, but he did have some respiratory problems. He was pretty normal size, but his biggest issue medically was low platelet count, about 100,000. I did have the petechiae, the little spots. I have purpura, by the way. Anytime I touch anything, I have purpura now at my age, so different problem. Anyway, he had to have a lot of transfusions. He actually did not have heart disease. Went on to have some delays and be short. And fortunately, I think the interesting thing, I guess, is down at the bottom, actually, because I still do sometimes look at fingertips, and he had multiple what are called arches. They're just these little tents on the fingertips, usually about a big long arch or whorl or something. He just had these little tents, and so does his brother. We'll get to in a minute. But I think his transfusion requirement basically went away once he was out of the newborn period. So what was the problem was, when his brother was born, we had not predicted that. We said, well, pure deletion syndrome, unlikely to happen again. But they went on and had a second one. So in his case, the platelets were exactly the same. He did have heart disease, the VSD, you'll see there in the middle. Undescended testes, minor anomaly, same as his brother. Short, so kind of a pretty typical case. And the fingertips issues were kind of the same thing, which is just kind of interesting to me. There are fingertip patterns that are sort of associated with syndromes, and I actually never thought about arches being overly prominent in this syndrome until I was looking back at this slide. And of course, you all know what the syndrome looks like. I pulled basically this off the site, so some of your kids may be on there, I suppose. Looks like most of them are having a pretty good time. So. That's good. So we do chromosomes by looking at them, like I showed you before, but we also do something called FISH. And this just happens to be a good FISH slide, which is of a different condition, but it shows what we're looking for. So in this case, the green dots are marking in the cell that there's a chromosome 13 in there. So if we were doing it for Jacobson, there'd be a chromosome 11 we'd be marking. And then the red one is actually the critical one, is that there on both chromosomes. So if we had a FISH probe for Jacobson syndrome, we'd be saying, we'd be expecting maybe that one of them will be deleted. Well, that's what's going on here. This particular gene on chromosome 13 is being deleted, called the RB gene. And so you basically count dots instead of looking at chromosomes. The cell's at a different stage. The chromosomes are all in there, all balled up, kind of like spaghetti, but you can find them by using a DNA probe. And why can you do that? Remember DNA? You can unzip it, create a probe, and then when it re-zips, the probe will light up the chromosome. So you put a whole bunch of probe in there, and instead of the chromosomes coming back together after you've opened them up, some of the probe gets in and lights it up for you. Does that make sense? So this matching property of DNA is just, it's everything. Okay, so when we look at FISH results for the family, so this is kind of a pedigree. So a square is going to be a guy and a circle is going to be a lady. So at the top are the parents, the blue dad and the pink mom. And they basically have normal chromosomes. They have this gene called MLL, which is down near where the Jacobson region breaks. It's present on their chromosomes, on the set on the left for each one. The set on the right for each one marks chromosome 11 again, and it marks what's called a telomere. That's the end of 11. So they don't have any breakage at the end. Those 11s look fine. So if we look at brother A, we actually found, doing FISH, that he has some normal cells. So, is there a pointer or no?
No, we've been using just the arrow on the computer.
Oh, sure.
Yours has it, but
Yeah.
there you
Okay. Yep. So basically, he's got some normal cells here, just like his parents. You see the blue dot marking the chromosome and the red dot marking the end. So these are not deleted. However, these are deleted, and his brother basically had 100% deletion. So none of his chromosomes lit up with this little red probe at the end. So we actually realized the first patient, and we may have done that before from chromosomes, I honestly don't remember how many we'd counted, but we discovered that he's what's called mosaic. So a mosaic is, basically you throw a bunch of colors together and match them up on a rug or whatever. In this case, you throw a bunch of cells together. You have normal cells and 11q deleted cells, and his body is a mixture of those. You don't have any idea what the percentage is in anything other than what you test. So we tested the blood, and I think it was around 10%. I'll get to that right in a minute. What's really important is, well, what's the percentage in his heart for heart disease? What's the percentage in his brain for development, etc. So basically, both boys had deletion, but one of them had about 10% normal cells. It was a mosaic. Parents were normal, didn't show any translocation. So we thought, well, maybe there's this mosaicism, which would be unusual to have 2 generations of mosaicism. But maybe there was a mosaicism going on in one of the parents, and that would have to be in the germ cells. So either the sperm or the egg that led to the boy's conception with a deletion, whereas the parent themselves is mostly normal. So mom was a little bit on the short side, had hearing loss and round face, and she was agreeable. Father was pretty devastated by all this happening. I mean, this was just really hard on him to have, first of all, 1, we're told probably won't happen again. Now 2, he cooperated, but he didn't want us to use any pictures. He didn't really want to get involved in any research, but mom did. Being more involved, I guess, in some ways. So she was graciously consented to a skin biopsy. So what we wanted to do is do chromosomes on a skin biopsy. So we basically looked for used it with a FISH probe, and we also looked, and she had about 20% deleted cells on a little piece of skin from her arm. So if we take the whole table, basically, mother is normal in the blood, so no abnormal cells. Father, down here, no abnormal cells. The first brother, remember, he's got some normal cells. Second brother doesn't have any normal cells. But here's mother, 70% normal cells by FISH and 30% deleted in her skin, or if we counted them and just looked at them, about 80 and 20. So those are almost always different for biological variation reasons. So a pretty substantial amount of mosaicism in her body. So basically at this point we're able to say, well, we have no idea what the risk is, but you certainly have a fairly high risk of having this happen again. And of course, with two children with significant difficulties, I don't think they were planning on having more children at this point anyway. I don't think there was a normal one there, if I remember. Okay, so maybe you remember this from high school, anybody? Good. This kind of goes with those translocations that I showed you too, with the 4 chromosomes. They could be A, B, C, D. This is about the same thing. This is basically, if you look at an inheritance pattern, this is what it could look like. So if a father has an A and a B and a mother has a C and a D, the kids could have these 4 possibilities, and they're drawn out by this Punnett square. And if you remember that, and if that makes sense, that's basically how we do inheritance studies in genetics. We just don't use A's and B's as such. We use various, what we call markers. So this is a whole set of markers on this family. So now we've got father up here, mother over here, brother A with the normal cells, brother B with not. So basically start looking at the end of chromosome 11, all the way down here, with all these markers to see what they look like. So we're basically able to tell the chromosomes apart. Whoops, I gotta go back one here. I didn't do it. Okay. Tell them apart. So basically, we're going to call father's chromosome this blue one over here, if I can find him. There he is. Blue and red is basically what father's going to pass on. So he's either going to pass on blue or red, or chromosomes cross over. So that's how we get a kind of a mixture of our mother and our father, the kid's grandfather, to them. So you can see here that this mostly the blue chromosome down here at the bottom, with a couple of little reds. So that means there was a crossover that happened right here. There's another one over here somewhere. We're not going to worry about the crossovers very much. What we're going to worry about is mom's chromosome over here. So if we look at her, we can differentiate the chromosomes. We can tell them apart because the numbers are different. So if you started at the top, there's a 166 and a 170. That's basically an AB. There's a 240 and 252. That could be a CD. There's a 141 and 149. That could be an E and F. Anyway, we can tell them apart. We can tell the 2 chromosomes apart. And we don't always find out. We'll find an A and an A every once in a while, and a B and a B, whatever. But if you look past the arrow, the numbers are actually identical on both sides. So her chromosome from her mother and her chromosome from her father, and we didn't have her parents to look at, are identical from that point down on chromosome 11. So we thought that was pretty unusual. And then when we look down at the kids, we find that the one boy, the second boy born, just basically has a deletion from here on. He has all of the markers from his dad, but none from his mom. And then here's his brother, who has all of these markers from his dad, but none from his mom, in a small percentage of cells, so an asterisk. So if you start here, there's a 147 on both, actually. Here's a 241 and a 237, tell them apart. There's the 241. Okay, your dad's got a 196 and a 287, there's a 196 and a 287, and on down. So we just don't see any of the mom at all in brother B below this point. And in the one guy, we see only dad. So something interesting biologically has happened here in this family. So we knew we could potentially write this up at this point, but we just didn't get around to it. I was trying to get Dr. Grossfeld's group actually to sequence the DNA here to find the breakpoints as to where these changes happen. But we did it a different way. So going back up to the basic structure of DNA, we've got two molecules of DNA at the top. One from a person A has got a blue and a gold strand. Person B is a yellow and a pink strand. Anyway, if we match them together again, most of the matches would be person A and person B to themselves. But every once in a while, our DNA, everybody in this room has a DNA that's 99.9% identical. However, the size of the DNA is such that there's hundreds of thousands of differences in that 0.1%. So that's why we're all different. And actually, if you look at a chimpanzee, we're like 99.8% identical to them, too. But again, there's hundreds of thousands of differences. So the differences are kind of shown down here as these little X's, mismatches. And a certain number of mismatches will be tolerated, and we can match up and see what we have. So So this is not a marker experiment looking at the numbers of the A's and B's and C's and D's. This is just looking at matching. So this is and this was a new test that came out, well, I'd say new, in the mid-2000s, and we were able to do this a few years ago and finally decided we should write this family up. So basically what happens is, on this little thing here, it's about the size of, let's say about half the size of a cover of a cigarette package. And it's got a little mirror in here, which you can actually, or a little piece of glass that you can see through. And that's probably no big, it's smaller than a quarter. Inside that is all these dots down at the bottom. Where am I? Here we go. All these dots here. There are actually 2.7 million of those dots in that little spot. And what those are is they're pieces of DNA from all across the chromosomes. They're only 40 bases long. We have 3 billion base pairs, but there are a couple and a half million of them. So they actually do a pretty good job of crossing our entire genetic material. So they're sitting on a slide, and so they're kind of diagrammed over here, standing up, ready to do something. They're all single-stranded. So we stick our patients in here. It doesn't matter whether it's DNA, RNA, that doesn't really matter. What matters is the matching. So in this case, there's been a match of these two. There's been a match of these. This is a different piece. And then all of a sudden, there's some extra matching this piece, and there's no matching this piece. Well, basically what this is showing you is when you do this matching experiment, you could say somebody's deleted for a region when they don't match what's on the slide. That would be this one. Or have extra for a region if they have too much matching, this being more normal. A little different way of looking at it, but anyway, so the spots are all down here. We're going to throw DNA on, and we're going to look for the intensity of the match. And so the way the computer looks at it is green is a higher intensity match, yellow is a normal match, red is a light match or a deletion. So basically you read all these dots off the computer. So this is what we got from Mom. So we had to put together 2 or 3 different things to kind of interpret what's going on here. So this is the software reading out what we just did. So basically what happens is we're matching these little points, 2.7 million of them, remember that, in this region, the chromosome 11, right over here on the right, that little piece down at the bottom, which is the deletion region for Jacobson syndrome. And we're showing a little extra up here, that white. So we just bracketed this region right here. So basically everything's moving along normally here. So we see all kinds of what we call noise. You know, the matching isn't always right on the line. It's a little above, it's a little below. There's a tolerability of, you know, it's just experimental error kind of stuff. There are some major deviations, and sometimes you wonder, well, what are these guys here, like that one and this one? And honestly, I think I checked them out, I can't remember what they were, but the important part is the whole baseline shifts down here. So there's less matching all the way across here. This actually I think was one of the boys. Same thing would have happened in the mom in a percent, same thing would have happened in the other boy in a percent. It was just easier to take the boy who was completely deleted. So basically up here is where the deletion is in red. You can see the baseline has shifted right below that. Right there. This is telling us that basically instead of seeing A and B together anywhere, we'll see either A or B. Everything is what's called homozygous. It's always the same. AA, BB, CC, DD. We never see C and D. We never see A and B. They're homozygous all the way across. And that's what this thing tells you right here with the 3 lines going down to 2. So this would be AA on the top, BB on the bottom, and AB in the middle. So everything's going along fine here until it hits here. Now there's no more AB. I mean, there's a little bit here and there, but that's just noise. So when we look back at our family studies with those markers, this one was still showing 2 copies in the kiddo. This one is only showing probably 1 copy, homozygous. Here's 2 genes. And here's a breakpoint called TNR number 5. That ought to ring a bell to you. Does it? It's been a while. Put him on the spot. An article by Jones. Took a bunch of your patients and tried to look at what are these breakpoints like. So there's this thing called a fragile site on this chromosome. Some of the patients seem to break at this fragile site. Fragile sites are areas of the chromosomes where they're kind of repeated segments of DNA, and they can kind of get mixed up and broken, basically. So what they looked for in this study was, are there other smaller fragile sites, regions of short DNA repeats that could be broken up? So the repeat number 5, called trinucleotide repeat number 5, is right there. And have you heard of the Fragile X syndrome? It's kind of what I'm talking about. So Fragile X syndrome, if you look at the X chromosome, it almost looks like it's breaking off. What's happening is there's part of that gene that has CGG, CGG, CGG. Once it gets to a certain number of times, it just kind of unravels and looks physically fragile. That's what they were looking for here. And they actually found a region of, I think it was only 6 CGGs right there. So that seems to be right about where our break point is. It's really very close. And then a fuzzier map, because I had to pull it off a copy of a copy, but this, you were co-author in this paper then. Where here's that break point toward the bottom. And how come my pointer finger just doesn't work here? There it is. Okay. So this was the last one. This was the one that looks like it's probably broken at. This is the last one we can see 2 copies of. This one we can only see 1 copy of, and here's TNR. So I bet the break was probably about the same. And this just describes the CGG thing here. CCG actually is what they had found. And a lot of standard language, but actually the language says that some of the other patients had breakpoints right there. So one of them was this patient 12, described in an article by Penny. So I don't know if any of your kids were that one. A patient RB in an article by Tunnicliffe. Somebody described in an O'Hare article, and 2 in a report by Lawrence. So there are actually at least these in the literature that look fairly similar to the ones we found. So. But certainly the breakpoints are different. There's those other TNRs that were found in that article you were involved in. There were patients broken all through there. I should go back to show these. These were the other breakpoints. You can see the yellow areas. Some of the other patients had broken there. Here's the fragile site I talked about. So what does all this mean? So why does losing one of 2 chromosomal regions cause Jacobson syndrome? So there are basically 3 possibilities. One is you have to have both genes present. You cannot get by with one gene. So that would be the simplest one. And we see this a lot in genetics. We see brittle bone disease. The movie Unbreakable had Bruce Willis, and who was the black guy? I'm still trying to remember his name. Yes. And he had osteogenesis imperfecta at birth and never liked the fact and became a murderer anyway. Not something most of my patients do. but anyway, one of the ways to have that is to be missing the amount of material and have just half the amount of collagen. So your bones are half as strong as they should be. It could be then that there's another mechanism going on, and that's what's called recessive disease. We've knocked out one completely, it's gone, and the other one has a mutation in it. Now that gene is non-functional. And things like cystic fibrosis have inheritance like that, but not necessarily this kind of situation. And then finally there's this imprinting, which actually our lab does the most of, or it still does in Montana, which is where you have to have the correct parental copy. So if you have one copy and you got it from the right parent, it doesn't matter that you're missing the other one. It really does matter in certain situations like this. So on chromosome 15, there's a real common area that can be deleted. And it causes a couple of different syndromes. So this is one. This is Prader-Willi Syndrome. And actually it was a CSI on a guy who ate himself to death at a hot dog contest because he got away from his group home. Those things happen with this syndrome. People just have an insatiable appetite. And they can be kept thin as long as you get right on top of it and keep food away from them, keep food locked up. Tough syndrome. So I was very confused when my friend told me that this until this was found out, the paternal deletion, that this syndrome had deletion in the same chromosome. I said, well, this is Angelman syndrome, and it's not anything like Prader-Willi. Well, it turns out that if you're missing the mother's copy, this is what you're going to have. If you're missing the father's copy, this is what you're going to have. So that's imprinting. So is there imprinting in this part of Jacobson syndrome? Apparently not. Because it doesn't matter which parental chromosome gets broken, the child still has Jacobson syndrome. So it doesn't seem to be imprinting, which is what I just talked about. It's probably not all recessive conditions. I mean, you have to have a bunch of them to get enough critical mass to produce a syndrome. So it's probably the haploinsufficiency. So some critical genes must be present in 2 copies. There is no evidence of imprinting, as I said. Some of the differences in one family to the next will be because of these recessive conditions that could be there. You just happen to be a carrier yourself, your partner's chromosome breaks, now your child has that carrier situation with no normal situation to counteract it. So they can have what's called a recessive condition. So what really happened in this family? Why were 20% of the cells in the skin of the mother and about 10% in one of the brothers normal? So it actually was the other way around. It was 20% abnormal in the mother and normal in the kid. Well, there is this thing called trisomy rescue that we learned about in genetics quite a while ago. It's actually involved, the first place it was found to be involved was in Prader-Willi and Angelman syndromes on chromosome 15. So they're not only deletion syndromes, but they're imprinting syndromes. So basically what's happening here is you've got an extra chromosome here. So you're supposed to have, so this basically is looking at the formation of gametes. So basically you're supposed to take a cell that's normal and split it into one pink and one yellow. So this would be normal. So you can see if you start with 3 here, there's no way you can divide it in 3 and 2 and get it normal. So sometimes you're going to send 2 pink ones, or in this case over here, you're going to send 2 from the same parent. So this is called Uni Parental Disomy. In this case, it's the same chromosome that's from the same parent. In this case, it's one of each from the same parent. So we think that's what went on here. We think that in mom, about 10% of her cells, 20%, that could have been deleted, actually were repaired from the other chromosome. So that's why I put this arrow here. And that would be called uniparental disomy, because now at that end of that chromosome, she's just got chromosome material from one parent. We never could tell whether it was her father or mother. They weren't involved, but we know by that test we just did that this is what's going on. So she could have been potentially a person with Jacobson syndrome who got mostly repaired. Some of her findings were probably because enough cells were deleted here and there and produced some of her issues: hearing loss, short stature. So I think there were some learning things too. And then the odd thing then that would predispose her, because she has actual deleted cells in addition to these cells. That would give her a risk to have another child with this, because she actually has deleted cells in her gonad. So what she's done in her blood is repaired everything. What she's done in parts of her body, she still has a deletion. What the kid did is he mostly has a deletion, but in some cells he repaired it, which is very interesting that 2 generations went on. This is just unheard of. That's kind of why the article went through when we submitted it, because it's just not unheard of to have mosaicism, and uniparental disomy. So there's something very interesting about the biology of this area that's able to repair itself. So before I go on to the next, this could be a mechanism for recurrences in those families that don't have a chromosome translocation. How would you find this? Well, probably the easiest way would be you'd be a little bit like this family, where one of the 2 parents has some signs of the condition, just enough so that it would be worth doing a DNA study to try to figure out if that's what's going on. Normally, we just say it's unlikely to happen again if there's no translocation. Okay, so just in the final few minutes, some things on genetics in general. So these are the genes that are in this region. And you can see it's a pretty staggering number, although a lot of these are repeated. And the reason they're repeated is they're copied differently for different cells. So one cell will copy this part of the gene, and the next cell will copy all the way over to this part, and another one will copy over to that part. Those are called transcripts. And they make our cells different from each other. So brain cells different than fingernail cells, for example. The genes are all the same, but the expression and the transcription has to be different. So if we look at all those genes, there are roughly 113 of them, depending on where you want to draw your break point. And a place to go to see what they might cause, if you're interested, is a resource called OMIM. It's called Online Mendelian Inheritance in Man. Dr. McCusick was sort of the father of genetics at Hopkins, and he put this database together personally by himself for years, Now it's been carried into the digital age, and he actually died a few years back. But if you look at all those genes, most of them we don't know much about. 17 we know something about, but only some of those are recessive conditions, and the rest are due to different kinds of changes in the genes. So recessive conditions probably don't play a big role in this syndrome. Then if we actually look at where the genes are, where the findings are, we can use that same map that I showed you before on the left. Okay, so here's a gene here that's probably involved in deafness in this condition, but there's actually another one. It's like I've got some type overs going on down here. There's another one toward the bottom right here. You've done some work on hypoplastic left heart syndrome, or VSD, was it? VSD or hypoplastic left with the ETS1?
Maybe all the above.
I think it's both, yeah.
It depends on the genetic background.
Yes.
Probably.
Okay. Yeah, and then the blood findings for the low platelets are probably here. The kidney findings can be here. Seems as though just looking at patients, if they're deleted above here, they're going to have finger anomalies. If they just have a deletion below here, they won't. So, you know, these are things geneticists play with to try to figure out what's going on in the syndrome. And there's really a lot more sophisticated ways to do that now. There's a project called ENCODE that looks at all the gene interactions. And if you want to take a look at an ENCODE slide, I should have brought you one. It looks like a giant spider web with about 80 flies caught in it. And each fly is a gene, and all the web interactions are with the other genes. It's so complicated. You know, 22,000 genes we've got, and they all interact with each other. It's going to be a while to figure that all out. One thing we have figured out using that technology I just showed you, where we could look at the matching process, is a whole bunch of microdeletion syndromes. So these are just the microdeletion syndromes from chromosome 1 to 11. If you look at the very bottom, there you guys are, Jacobson syndrome, right there. If you finish them off, there's all these. And these are just sort of major recognized syndromes. And I'd say at least half of these have come in the last 5 to 10 years since that technology was developed. And then it turns out there's microduplication syndromes. And you can see most of them are also microdeletion syndromes. So it just keeps getting crazier and crazier. So when we see kids with autism, the test we do is the one I showed you, the really fancy slide of with all the lines and the matching points and all that. And in about 20% of kids with autism will find something on that test. But if you look at how many kids have the same thing, very few. So some of them actually have some of these conditions, and that's good because there's support groups for some of these conditions like there are for yours. So you no longer have to feel very lonely when you look at this slide. There's a lot of stuff out there for people. But it's also getting more and more complicated all the time. So the TNR things that Dr. Grossfeld's group looked for years ago have been somewhat supplanted with these blocks. So you see these colored blocks here. Looks like a kid's Lego set. But this is actually talking about chromosome 22 and how it breaks at various spots. And the reason it breaks is because there's this repeated DNA blocks through it. So you see this yellow-green, yellow-green, yellow, yellow-blue down here, yellow-blue. A couple of these are identical. So chromosomes are trying to line up and exchange material. But if they happen to misalign just a little bit, these blocks will allow them to change material. Then one chromosome's deleted, the other's duplicated. That's why for almost every deletion syndrome, or a duplication syndrome, just usually takes us longer to find them because they're not as severe. So, in summary, I've told you about this family. It had a recurrence. Recurrence was due to mosaicism in the mother, and that had to do with the deletion part. But in all of her blood, and who knows what in her tissues, she had repaired a deletion. So she actually started out with a deletion probably everywhere. So she ended up with a deletion about 20% left in her skin, and obviously the ones in her ovary caused the recurrence. We know that this kind of conversion of a break to normal, it happens a lot in cancer, but we're not really as familiar with it in syndromes like this. The flip side, so this is a deletion, what we call rescue by repair. The flip side of it, that trisomy rescue, is pretty common in reproduction issues. But the deletion rescue occurred in this case, making the mother's findings less severe. Okay. Anyway, I don't think this is terribly uncommon. I just don't think it's been looked for or found that much. And in terms of you guys, in terms of relevance, I guess I would just say that if there is a situation that comes up in your family whereby you can see where one of the one of the parents of a child looks a little like the syndrome, you might want to think about this happening and potentially present a different recurrence risk. And of course, anytime it does recur, I'm sure it was tough. It's just tough. I mean, to end up with 2 of these kids, as much work as it is with one, I don't know how you do it, but hopefully they're good kids for you. Yeah. I mean, and it makes you wonder, what's wrong with me? I can't have a normal kid. Well, in some cases, some crazy, bizarre genetic mechanism has happened to you, and that's why. So you can't feel bad about it. I mean, it's no worse than me being delayed in DFW yesterday for 5 hours. SHI happens, right? So, okay. All right, well, thanks for your attention. I'll be around for a while. Appreciate it.
Do you have any questions?
I do, if no one else does.
Thank you for being here, Dr. Jonathan. It was very informative. I have a question, and I was wondering...
Where are you? I'm sorry.
Oh, I'm over here, next to Lindsay.
There you go.
Do you have any idea what role folic acid, like the mother taking folic acid prior to pregnancy, could have? Because we weren't planning to get pregnant so early, so I was not taking folic acid, and I always, it's this guilt thing that I have. Would that have prevented this?
It's like getting laid up in Dallas. It's just one of those things. At least 50% of women who get pregnant have been drinking, like the day before.
Right. I was probably in that kind of...
Yeah. Yeah, I mean, you're out on the ship partying and you get pregnant, right? I mean, that's the way it happens. How many fetal alcohol syndrome kids result from that? Probably 0.
Right.
And we see women who've had a child with a cleft lip and palate. That's where folic acid has its biggest effect.
Okay.
They didn't take it, their child has a cleft, they feel bad, but for every one of them, there's 10 that took exactly what they were supposed to, maybe even more, and still have a cleft. It's just too complicated.
Yeah, it's one of those things you're always kind of looking for, like, well, what if I did this?
Exactly.
Or that, or what, actually.
And we always try to tell people, it's just not that. I mean, there's just so many factors involved. It's just impossible in any case to pinpoint them. Did radiation cause your breast cancer? Possibly. But, we know it does in 1 out of every X people. But are you the one?
Right.
We don't know. Yeah.
A little trivia to add.
Yeah.
We actually found that in most cases, the break point does not cluster around a fragile site.
Right.
That's number 1. Number 2, we've done a race CGH now between here and in Italy with Dr. Mattina and probably well over 100 patients. And it's actually incredible how it's almost a completely random distribution where the break points are. So there's almost no clustering, which suggests there are other mechanisms. In Laura Penny's paper that she published back in the 90s, she actually showed that there is, when the deletion is up to a certain size, in all cases, the deleted chromosome comes from the father. So there is some kind of...
there was...
Yeah, and I'm not sure what to make of that. One possibility is maybe if it comes from the mother, that's lethal for the fetus, but... So...
Do you know what that size is?
Yeah, I'd have to go back and look. I think it's a marker called D11S924.
Probably on my map.
Yeah, it was on your map. I think that's the one. There was something...
but it's not unique. There's a favorability for bigger deletions coming from, did you say dad?
Smaller deletions.
Smaller from
For the
dad.
smaller deletions, at least 10 out of 10 that Laura looked at, it was always derived from the fathers.
Since then, the numbers have changed a little bit. There's a preference for proximal deletions being one parent and distal further down being the other, but it's not 100%.
And I don't know what to make of that. So here's a practical question that comes up not uncommonly, and in fact there is one family whose name shall go unmentioned that I'm actually trying to help right now, who has a child with Jacobson syndrome, and they're thinking about having another child, and they want to do everything within their power to ensure that there's not a recurrence.
Okay.
Both parents have had chromosomes tested from peripheral blood lymphocytes and they were normal.
Right.
So then the question is, what about the possibility of mosaicism?
Right.
And so one possibility in theory, you can't go and test the mom's eggs because you'd have to harvest her embryo or her ovaries.
Yeah.
You could in theory test the father's sperm and determine if there's mosaicism, although I have not found a CLIA certified lab that can do that. I don't know if you know of any. So another possibility I was thinking of though is knowing that I know of 3 cases around the world, so about 1% of all families in which there's been one child with Jacobson syndrome in which both parents test normal by looking at their blood cells, there's been a recurrence. So that brings up the idea of this phenomenon of germline mosaicism. So one thing, actually, after reading your paper, I thought is, what if you did a skin biopsy and proved that there was not mosaicism in the skin cells? Then you have really two lines, blood cell lines and skin fibroblasts, which I think would even give you more reassurance. And then, of course, you obviously weren't here earlier in the week, but we heard a very elegant presentation by Charles Cantor from Sequinome about the maternal blood analysis of fetal DNA. So, any thoughts that you might have about? Yeah. That's too much rambling on my part, sorry.
No, that's fine. I guess if you're going to go to the point of doing a skin biopsy, you might want to do this either by doing an array or by doing the marker studies that we did to show that there's not been a repair of a deletion that could be elsewhere. Maybe you don't see it in the blood or the skin, but you actually see this repair effect that's happened. I mean, the best way to do it is just a matter of, it all comes down to money, is the pre-implantation diagnosis. Right. It's really pretty good with chromosome markers. So you can use the fish technique I showed where you're counting dots. And you can put together some embryos, count the dots. If the 211 dots are there, those are the ones you put in. If they're not there, you don't put those in. The person typically is going to, the woman's typically going to have at least twins, maybe triplets, because they always want to put a couple in to make sure it's effective. And it's about 99 point something accurate. It's not 100%. But it's also probably about $20,000 to $30,000. But some insurances will pay for it because you make the argument, we don't want to have this happen again. This is expensive. Congenital heart surgery in the first week, it's way more than $20,000 or $30,000, yeah.
One point, let me just finish one point and then we'll come to that. So pre-implantation diagnosis, as he mentioned, is a way to prevent a recurrence by screening a couple of cells from a fertilized egg at about maybe the 16-cell stage. Now, it depends where you read this, but in this case, since we know that in that family situation, the chance of recurrence is probably no higher than 1%, I've actually kind of suggested not doing pre-implantation diagnosis because it's institution user dependent, but there can be a higher frequency of chromosomal anomalies in general arising from the actual technique. And so, for example, what if the chance of having any chromosomal abnormality from pre-implantation diagnosis is 5%, then you're really doing yourself a disservice versus 1%. So I've actually recommended to them that they do not pursue that in this case.
Well, yeah, and actually our lab spends a lot of time looking at in vitro fertilization, and there are some imprinting defects related to in vitro fertilization too. So there's certainly a lot more testing, though, you can do in that embryo than just the 11. You can try to get it right. But.
Is there any way for a parent to have their DNA tested to see if it explains why they have a child with 11q or a child with Jacobson syndrome to determine if they want to have, if they're at risk for balance translocation or other reason?
Yeah. Well, conventionally, yeah, they should have chromosomes done to look for the translocation. And if you want to go a step further, based on what I'm saying, you could do a chromosome microarray on the parents, and you would see if there's been this correction issue that's gone on, at least in the blood, which is typically what you look at. Probably wouldn't necessarily go any further than that, because recurrence is not common in this condition.
And typically most most insurance won't pay for it, right? So you have to get the healthcare to pick it up.
Yeah, they would pay for the chromosomes. The microarray, you'd have to take my article and say, this is why you ought to do it.
Well, that's why we publish things, though, right?
Yeah,
That's why he's here. I have another question for you. As far as the mechanism, this is really looking into the future, but as far as the actual mechanism of uniparental disomy, I think it's fascinating that somehow a cell has the machinery to actually repair a deletion.
Yeah.
And forgive my ignorance on this, but what's known about that machinery, how that works? How does a cell sense when there's been a deletion, and what initiates the process? Does anybody know? I'm sure that's been looked into.
I'm sure it's been looked into too, and I can't exactly answer it for you, but on a smaller level, it's involved in something called gene conversion.
Right.
And gene conversion is really where gene therapy is trying to go to basically take the normal gene and copy it into the abnormal gene and fix the abnormal gene. Although I got to say, gene therapy's been a lot of hoopla and a fair amount of disappointment over the last 25 years. And where it's headed now is more looking at what the genes are doing. So we need to know those genes in this area, what they do, and which ones are most important, and then pharmacologically, what can we do to alter that gene function to make it more normal.
And that's a lot of what we've been talking about for the last couple of days.
Yeah. Lunchtime?
It's lunchtime. Thank you again so much.
What's the time frame?